The Antarctic, one of man's many unexplored domains. Today we focus on the most predominant species of fish residing here. Come with us to discover the mysterious Antarctic Blackfin Icefish, scientifically known as Chaenocephalus Aceratus. With its unique black fins and icy habitat, it's truly a wonder of the sea. But what's more interesting about this fish is its evolutionary ability to live in such cold environments. Dive into the chill with us and explore this revolutionary adaptation today.
My name is LYNDERA, and I am here to introduce our fascinating creature of the day, the Antarctic Blackfin Icefish, or Chaenocephalus Aceratus. Sorry, I like pronouncing it that way. As we delve into the fascinating world of the Antarctic icefish, we will be exploring its evolutionary history, unique adaptations, cellular mechanisms, and ecological significance. The Antarctic blackfin icefish, Chaenocephalus aceratus, belongs to the family Chaenocephalus. The blackfin icefish inhabits the cold, icy waters of the Southern Ocean surrounding Antarctica. Specifically, they are found in subantarctic and Antarctic regions. The blackfin icefish seems to like diving deep, but they are typically found at depths ranging from approximately 330 to 2,600 feet. And a fun fact before we get started, unlike most fish species, the Antarctic blackfin icefish lacks the pigment responsible for coloring its blood red, which is hemoglobin. Rather than hemoglobin, the Antarctic blackfin icefish possesses a translucent, nearly colorless blood due to the absence of erythrocytes, which are the red blood cells present in almost all vertebrates. So this unique trait has earned them its moniker, white-blooded fish. The Antarctic blackfin icefish, or Chaenocephalus aceratus, has a diverse diet consisting primarily of krillfish, which are like a shrimp-like crustacean. I don't know if you've ever watched Happy Feet, but I'm pretty sure they were in Happy Feet. But its diet consists of the krillfish and a various of small invertebrates that inhabit frigid waters of the Southern Ocean. The physiological feature that we will be focusing on today with the Antarctic blackfin icefish, or Chaenocephalus, or scientifically known as Chaenocephalus aceratus, is its antifreeze protein that allows it to adapt in freezing cold temperatures.
And my name is Jaheim Wallace. As Lynn mentioned, the Antarctic blackfin icefish inhabits the frigid waters of the Southern Ocean surrounding Antarctica, with temperatures of nearly below freezing points of -2 degrees Celsius. These waters are characterized by its high salinity due to the freezing and melting points of the sea ice, creating challenges for marine life. But what sets the Antarctic blackfin icefish apart from these other marine life is the antifreeze proteins in its blood. As we take a microscopic look with ice with and without antifreeze proteins, the one on the left is growing normally in water, but the one on the right that contains antifreeze proteins is binding to it, arresting its growth. These same proteins bind to the ice crystals in the fish's blood, and it keeps the ice from growing and freezing the fish. I will now allow my colleague Destiny Ernst to explain this on a more cellular level.
As LYNDERA stated, the Antarctic blackfin icefish possess these antifreezing molecules known as glycopeptides that are synthesized by the liver, distributed into the bloodstream, and makes its way into the extracellular fluid of the fish. Any other fish without these antifreeze glycopeptides would literally freeze to death in these extreme water temperatures, which makes this mechanism so mind-blowing. The glycoprotein surrounds the ice crystals that begin to form in the fish's body fluid and prevents the ice crystals from growing any bigger to the point of cellular damage or even death. The thermobiology of the Antarctic blackfin icefish also plays a significant role in the fish's ability to regulate its body temperature in these extreme water conditions. The freezing point of the water itself is negative 1.9 degrees Celsius. The normal freezing point of the Antarctic Blackfin Icefish is negative 1.2 degrees Celsius, which is below the freezing point. However, the antifreeze molecules increases the freezing point of the fish's blood to negative 2.7 degrees Celsius, which is above the freezing point of the Antarctic seawater. If that isn't mind-blowing to you, I don't know what is. Think about it. The freezing point of the fish's blood is below the freezing point of the water that these fishes live in, which means that these fish are at risk of freezing to death every second of the day. But when these glycoproteins are released into the fish's body fluid, they increase the freezing point of the fish's blood until it is above the freezing point of the water, allowing these fishes to swim and live and eat happily in these ice-cold water temperatures. Isn't that amazing?
The adaptation of antifreeze blood cells in the Antarctic Blackfin Icefish is a remarkable evolutionary trait. This has allowed these fish to survive in extreme cold waters in the Antarctic. The evolutionary significance of this adaptation lies in the fact that this allows the fish to prevent the formation of ice crystals in their blood, which could be lethal in such frigid conditions. The presence of the antifreeze blood cells in the Antarctic icefish is believed to have evolved as a response due to the extreme low temperatures in their habitat. These fish have a unique protein in their blood that acts as an antifreeze, preventing ice crystals from forming, causing damage to their cells. This adaptation has likely developed over time through natural selection, where individuals with the trait had survival advantage and were able to produce and pass this gene on, and which is responsible for the antifreeze blood cells in their offspring. Climate change has played a significant role in driving evolutionary changes in the Antarctic icefish. As global temperatures rise, the Antarctic environment is also experiencing changes, including warming waters. The shift in climate has put a pressure on the icefish to adapt to changing conditions, including variations in temperature. But this adaptation of the antifreeze blood may have become more crucial for the survival of these fish as they face new challenges brought by the climate change. Scientists have identified many causes of this adaptation to be a specific gene that has codes for the antifreeze protein in the blood of the Antarctic icefish. Through genetic studies and analysis, researchers have been able to trace the evolutionary trait of this gene and understand how it is able to allow the icefish to thrive in their icy habitat. In addition, the antifreeze blood cells in the Antarctic icefish has also evolved structural buoyancy adaptations to survive in their environment. These adaptations include reduced balloon density, lack of the swim bladder, and a unique body shape that allows them to navigate efficiently in water. This diversification of structural buoyancy adapts in the Antarctic icefish has further contributed to their ability to thrive in the extreme conditions of the Antarctic. Overall, the adaptation of the antifreeze cells in the Antarctic icefish is a remarkable example of the evolutionary change driven by environmental pressures and natural selection. This adaptation, along with the structural buoyancy adaptation, has allowed the icefish to survive and thrive in one of the most harshest environments on Earth.
Okay, so we will be talking about the significance of this physiological factor to the Antarctic Blackfin Icefish. Firstly, we will be talking about the importance of the chemicals that cause the Antarctic icefish to produce its antifreeze proteins and how they react. With the antifreeze proteins in the Antarctic icefish, they function by binding to ice crystals and inhibiting their growth. So this causes the prevention of formation of ice within the fish tissues. The chemicals or molecular structure critical for the antifreeze proteins to function effectively is the ice binding site, also known as the ice binding face. This site typically consists of specific amino acids, residues arranged in a particular conformation or pattern that allows them to interact with the surface of ice crystals. These amino acids, they form hydrogen bonds with water molecules in the ice lattice, which disrupts the regular crystalline structure and preventing further ice growth within the tissues. The ice binding site and the amino acid residues, they vary among the different types of antifreeze proteins, which gives them unique ice binding properties and allows them to function optimally under different environmental conditions. So with this allows the Antarctic black icefish to survive in its rather cold habitat because its ice binding sites of the antifreeze proteins, they contain the amino acid residues that are specific and well suited for the binding of ice crystals. Next, I'll be talking about how important the adaptation is to the future of biomedical research. And while the importance of the icefish's antifreeze proteins is important, it has not yet been fully realized, and their unique properties make them promising candidates for future applications, such as cryopreservation, organ transplantation, further like biotechnology and medical imaging. In my opinion, the most important of the four is cryopreservation. And it's also pretty interesting because the antifreeze proteins, they have the ability to inhibit ice crystal formation and growth at sub-zero temperatures. The blackfin icefish antifreeze proteins, they could potentially improve how cryopreservation of cells, tissues, and organs is done by reducing ice damage and improving cell viability during freezing and thawing processes. And that process in turn makes it easier for organ transplantation to be more efficient. And lastly, I will be doing a comparison between the Antarctic blackfin icefish, or Chaenocephalus aceratus, and its physiological adaptation in comparison to other marine animals that live in similar climates that do not have this adaptation. The antifreeze proteins that are found in the Antarctic blackfin icefish provides a distinct advantage over other marine animals in the same environment that lack the same proteins that are found in the Antarctic blackfin icefish, particularly in coping with the challenges posed by freezing temperatures in their environment and their habitats. While other animals have generated their own physiological adaptations to assist in surviving extreme temperatures or extreme freezing cold temperatures of sub-zero temperatures, the Antarctic blackfin icefish has its unique ice crystal inhibition. And I have already explained that, but it's when the proteins, the antifreeze proteins, they specifically bind to ice crystals and inhibit their growth. Specialized physiological adaptations that complement the action of the antifreeze proteins. This is reduced skeletal density, increased lipid content, and modified muscle composition. So all of this contributes to their buoyancy regulation and cold tolerance. They work hand in hand with the antifreeze proteins to enhance their ability to survive in the Southern Ocean.
Here's a fun fact. The glycoproteins are not released into every single body fluid of the fish, or at least not in high amounts as they are in other body fluids, such as the peritoneal fluid, pericardial fluid, and extradural fluid. Some of the body fluids that the glycoproteins are not released into or not in high amounts are the urine, endolymph, cerebrospinal fluid, and semicircular canal. And this is because many of these fluids already have barriers that prevent ice crystals from forming in these fluids. For example, ice crystals cannot enter the urethra of the fish due to the closed urine sphincter that is lined with a lot of mucus. Additionally, other fluids like the endolymph already have surrounding tissues that contain glycoproteins, which protect the endolymph from forming ice crystals. Therefore, the endolymph itself doesn't need the glycoproteins because it already has the glycoproteins surrounding it in tissues. In conclusion, the Antarctic Blackfin Icefish is truly a wonder of the sea, with its ability to withstand freezing cold Antarctic waters due to this physiological adaptation of antifreeze glycopeptide molecules, which inhibits the growth of ice crystal formation inside the fish's body fluid, resulting in them freezing to death. The icefish is able to thrive in conditions that other fish species simply cannot. Over the years, these fishes have naturally adapted to these waters, and natural selection is the only answer as to how exactly they were able to develop these antifreeze proteins in the first place, which makes us wonder if other fishes would be able to adapt to these temperatures with and without experimental manipulation if placed in these conditions, and if so, how long would it take for them to adapt? Along with these glycopeptides that contribute to the thermal biology of the fish, its specific ice-binding sites that contain amino acids, and the structural buoyancy of the ice fish, skeletal density is reduced, lipid content is increased, and muscle composition is modified, which all work together to make it easier for the antifreeze molecules to work efficiently. We hope to see more work being done with cryopreservation, organ transplants, biotechnology, and medical imaging using these antifreeze glycopeptides to improve cell viability. Thank you for taking the time to watch our video, and I hope that you've learned something along the way.